I'm Ryan Howard. I'm a staff scientist here at MRIGlobal, and my co-author is Megan.
I'm Megan Lemon. I am a staff scientist in Gaithersburg in IDSD.
And today we're going to be presenting our poster, Evaluation of a Targeted Next-Generation Sequencing Diagnostic Assay for Identification and Characterization of Drug-Resistant Mycobacterium tuberculosis isolates, which is a mouthful. But really what this poster is and this project kind of was a bit of a consumer report for a diagnostic assay as part of our NIH TBQA program. So what we did is we took an assay that's gaining some traction in the tuberculosis community. It's kind of an up-and-coming technology, and so as part of the program, we advise clinicians, policymakers, other stakeholders of best quality practices or assessments of new and upcoming technology like this. And so what we decided to do was to use our existing tuberculosis repository in collaboration with Johns Hopkins University Hospital. And what we did was we used known, well-characterized isolates of tuberculosis and then ran them through this assay to kind of evaluate it, put it through the paces, and get a feel for what it can do, maybe what some of the gaps are, and overall ease of user friendliness. What this assay does is we're looking specifically at antimicrobial resistance. So being able to know that if a patient comes in with tuberculosis, what is the best drug treatment regimen to give that patient? Because more and more we're seeing that people will come in and the traditional antibiotics like a rifampin or an isoniazid won't treat the patient. You can give it to them, as much of the drug as you want in it, and it won't necessarily be effective against that particular isolate that the patient's infected with. And so we see this is very similar to kind of the in the news as far as SARS-2 with these spike protein mutations, right? It's the same thing in Mycobacterium where you have a mutated gene, and if you remember the central dogma of biology, that mutated gene will lead to a mutated mRNA, which leads to a mutated protein. And so in the case of tuberculosis, a mutated protein such as RNA polymerase, for instance, would normally, you would give like rifampin to a patient. That rifampin would bind to that protein, inhibit transcription, and halt the disease in its tracks. But with that mutation, you get a little bit of an altered binding site. The drug is no longer effective, and suddenly you need something else to treat this patient, or else they're going to die, right? So the whole kind of reason why this is being looked at is for the end patient, and in looking and making sure we know, tuberculosis is exceptionally complex. A rough average of maybe 15 different antibiotics can be given, and we have in our current databases right now somewhere around 45 known genes that can give resistance to any one of those 15 or so antibiotics. So it's a very complex system of trying to treat each individual patient. And it's also very expensive to do this kind of testing, so you want to use the best assay available and make sure you get it right the first time. And so this assay is a step in the right direction for that, and we wanted to evaluate it. So I'll let Megan talk about exactly how we went about comparing and the materials and methods.
Thanks, Ryan. So one of the things that makes tuberculosis testing very complicated is that tuberculosis is very, very slow growing. So it can take 6 weeks to get results if we just grow it by traditional methods. And so that makes diagnosis and finding the right treatment very complicated if we just rely on phenotypic methods. So there's been a big shift toward focusing on genetic evaluation, as Ryan was saying, to speed up that process. So we had 3 different platforms spanning both phenotypic and genotypic evaluation, in addition to this D-plex assay. So the gold standard in clinical diagnoses has been the midget tube, and this does rely on that phenotypic gold standard of growing bacteria in the presence of different antibiotics and seeing if it grows or does not grow, thereby understanding its resistance profile. And that does take, up to 6 weeks. So that's, not ideal, but, does give us, a, a, a good feel for whether a drug is effective against it or not. We also did a line probe assay called the Bruker-Hain genotype, MTB-DR plus, and the SL, which looked at some first and second line drugs, and these were much quicker. The line probe assay relies on using PCR product and binding to a strip. And so we can get some information in as little as 8 hours with that. And then the last was the Cepheid Gene Expert. It's a self-contained cartridge, very, very easy to use, very little training required for that. And you can get antibiotic resistance information only for rifampin in under 3 hours. So the last 2 are very, very quick, but give less information. 7 drugs for the line probe assay, only 1 drug for Gene Expert, and I believe 4 drugs for the midget tubes. So GenoScreen was very, very interesting because it not only diagnoses whether this is tuberculosis or a non-tuberculosis isolate, but it also gives drug information. It can give lineage information. It's supposed to promise a whole lot more information in this assay. So that's really interesting, really something that we wanted to take a look at. So these the isolates that we used, we had 14 tuberculosis isolates that were previously characterized. We had drug resistance information about those isolates, and we wanted to compare those to what the D-plex assay was going to give us. But we also wanted to throw in some non-tuberculosis isolates, and we had 15 of those, and all 15 were correctly identified by this assay, which was pretty impressive. So the isolates are grown up on LJ media, a solid media, and then the DNA was extracted, and then we followed the manufacturer's protocol, taking this from extraction through PCR, proprietary PCR, then through library prep and sequencing, and then the data from the sequencing goes into the D-plex database for analysis. And we had, we had 13 first and second line drugs were evaluated in the D-plex assay, and all of the processing takes about 3 days to get through extraction and PCR, library prep, and sequencing to get it into that database. And from that, we got a lot of information, but wanted to compare that to what we had from our previous characterization of our strains. And with the exception of isolate 54 there with the Expert Ultra, everything matched up. So that's pretty good for the performance of the D-plex versus our standard other methods that we're using. And from that, I'm going to let Ryan take over talking about the bioinformatics portion of this.
Yeah, so we compared not only to standard, maybe more cost conscious diagnostics. I think the Gene Expert is something on the order of $9 or $10 a cartridge, subsidized by foundations like the Gates Foundation or other stakeholders. This sequencing isn't as cost competitive, but it also provides a whole lot more information, we think, than some of these more limited diagnostic means. So you see here, for these other diagnostics, we were able to compare, two drugs, and some of them, have more information, but we couldn't do a whole, characterization of everything that's output by the D-plex assay to just one or another commercial diagnostic. It's a lot more than that. So if I zoom in here, you can see, here we're getting, this is an output table from the D-plex web app, and you can see all these different drugs here that are being screened, looking at known genetic mutations in bioinformatic databases, and seeing, okay, was there a single nucleotide polymorphism for isolate 35 here? It looks like yes, and that mutation is known to cause resistance to rifampicin, right? Or over here we see with isolate 67, you see just mutations across the board here. This is what we would call a extensively drug-resistant strain, and you'd be really hard-pressed to find, if you didn't know this beforehand, you'd be hard-pressed to find an antibiotic to give a patient with an isolate or a disease isolate like this. You'd really be stuck with some of the very newest drugs that are typically only used in certain cases when, as a last resort, when it's known that, okay, we don't have anything else that can really treat this. We're going to, bring out the big guns like a linezolid or a bedaquiline, clofazimine. But as far as the bioinformatics portion, I'll shift over here. This is kind of a figure of some of the web output. And what you can see here is a kind of a graphical display of everything that was just in that ugly Excel table. You can see, for instance, each region that's targeted. You can see the overall coverage and the particular mutations that were identified for a given lineage or a given isolate. As well as in the middle here, you can see a lineage or a spoligotype that's been identified. This can be used for potential outbreak tracing, epidemiology. We didn't really go into testing that. Most of our isolates in the TBQA repository are not related, so we'd be hard pressed to find data that we could really evaluate that with. But it's another interesting feature of this assay and web analysis. And one thing I keep saying is web analysis, and I think it's important to point out that once you do the lab work and you do the extraction, the library prep, put it on the sequencer, after that you're really pretty much done. You don't necessarily need a trained bioinformatic staff member to evaluate this assay, and that's unique among a lot of the next-gen sequencing assays. It's a web portal where you just upload your DNA, and then all the analysis is done automatically, and it spits out a report using colors that show, okay, if something's green, that means it's believed to be susceptible to that particular antibiotic. If it's red, there's resistance there, and that shouldn't be necessarily maybe used to treat a patient, and it's giving guidance on that treatment regimen. You can also see here, for instance, so we had a little discrepant data as far as for certain second-line injectables, kanamycin, amikacin, et cetera here. And you can see that we didn't have actually a lot of data. We had some what we suspect to be primer dropouts, and you can also see that here. So if I zoom in here, this is the coverage representation, and you see that, okay, for this particular isolate, we had pretty good coverage for everything, but there's a definite dropout here where we're not getting any information. and that corresponds to those antibiotics. So it's not necessarily maybe a perfect assay right now. I think there are definite improvements that could be made. It's not like we saw this dropout on just one isolate, right? It's the large majority. For some reason, there's a dropout there, with only a few of them actually coming through with enough data to be able to recognize if something's resistant. So that's something we're interrogating more. But overall, again, going back to this, consumer reports mentality, I think I'd give this 4 stars. I don't know, Megan, any star ratings?
That depends on how fast you want data, how much data you want. I think it really just depends on what your application is. If you want a quick and dirty, does this patient have tuberculosis and is it susceptible to rifampicin? The gene expert is definitely the way to go. Cost effective and quick and easy. But if you're looking for more specific information and you want, a full panel of drugs and you have skilled technicians and time, a couple days, then this is definitely a lot more information, more bang for your buck, I guess. Although it is a lot of bucks, so there's that too.
But I'll add, it is also, I think, a lot more efficient than like a whole genome sequencing, where you're sequencing 4.4 megabases of the genome for each isolate, right? This is a targeted, we only want to know these 18 primer regions, and that's it, and we get a lot of depth there, and we can multiplex up to 45 or 48 isolates on a single MySeq cartridge. So it becomes a lot more cost efficient as well for the information that you are getting.